So let's go ahead and hi everyone again. So we have had a short break, and I'm more than delighted to introduce our annual keynote. And This year, this is Professor John McGann from Rutgers University. He is director of Rutgers Center for Cognitive Science, and he's also director of the lab on neurobiology of sensory perception. His main field is to understand how brain transforms sensory input into coherent information, and the focus in that area is olfactory perception and how olfactory receptor neurons are operating. We all know that COVID-19 side effect is anosmia. So some of us have done studies on anosmia, and well, this is one way how to see how important actually olfactory perception is. And Professor McGann has received numerous grants from National Institute of Health, from National Institute of Deafness and Communication Disorders, and is among the most prominent colleagues working on neuroscience of olfactory perception. I have to say another more personal remark. There is a publication, 2017, science publication that was groundbreaking, not just in my case, but also lots of colleagues working on the psychology of perception were really kind of experiencing renaissance of olfactory perception, because the title of the paper by Professor McGann is Poor Human Olfaction is a 19th Century Myth, which is true, because we know that human olfaction is not just a powerful sensory processing system, but is also a system that processes emotions, because olfactory bulb is a part of our prefrontal cortex, is linked to memory, and well, not last but not the least, humans are incredibly powerful in ability to discriminate among scents, which is something that is even stronger if you compare vision or auditory perception. So anyway, without wasting any more time, I'm more than delighted and honored to give the floor to Professor John McGann.
Thank you very much for the intro, and I really appreciate the invitation. So it's funny, based on the body of work I've put together over the last few years, I now seem to be frequently introduced as somebody that studies smell. And it's true, I admit it. But I didn't start out intending to study smell. It was a venue for me to try to use a sensory system to try to understand the mind, to see how learned beliefs, expectations, things that we understood about how the world worked would actually manifest in a sensory system in a case where we potentially already knew the brain's code for the outside world, and we could potentially understand how the neural activity could relate to things like beliefs and learning and memory and things like that. So I'm going to start by giving you a little bit of an introduction to smell. And since I'm going to show you data from both humans and mice, I'll give a little bit of a warm up to this idea that humans have a good sense of smell too. And then we'll get into some data that's mostly neurophysiology for a while. So let me start just saying that smell tells all of us about the world. We've got a huge range of critters on this planet, and essentially everything from single-celled organisms to us. We all use chemical detection to understand what's happening out there across a whole range of animals, including professors. And the moment of smell in our lives, we often don't pay as much attention to it as we could, but most of what we experience as the flavor of our food is in fact odors going up the back of your throat and reaching your nose. And that applies both for wonderful tasting things like spices, but also for some more complex, sometimes unpleasant odors that shape our eating and also our experience walking around in the world. But it's more than just food. It turns out that Odors actually have kind of a remarkable ability to just help us decide whether to eat. So I don't know about you, but I've never gone to a movie theater to get popcorn. But if I'm in a theater, somehow I always end up eating popcorn. In the US, we also have this bakery chain called Cinnabon that sets up in malls and pumps these beautiful cinnamon bakery smells out. And then somehow I never intended to eat Cinnabon, but then I'm navigating a course through the mall and I'm taking green paper out of my pocket and I'm handing it, and then I'm eating food that a stranger gives me, all triggered by a few molecules that somehow got into my nose, right? So it helps us decide whether to eat, but it also helps us decide what to eat. And so here's a picture of a man's sense of smell potentially saving his life. I didn't realize for real until I had COVID over this past winter holiday that I apparently almost eat a lot of things that could be spoiled. And I found myself reaching into the fridge and going, I can't tell if I can eat this, right? And of course, the cultural differences in what foods are considered delicious reflects the plasticity that our sensory systems experience, not just preference, over the course of our lives. Let's move on to this idea that Jurgis alluded to, that humans and mice both have an excellent and an impactful sense of smell. So I'll just hit a few highlights on this. This is a half of a mouse's head, and you can see the air comes in from the nose and it flows over these turbinate bones where the olfactory epithelium is located. We'll talk more about that in a little while. And the axons from these receptor neurons in the nose, they go back to this big structure here called the olfactory bulb. In a mouse, it's out in front of the rest of the brain in a sort of prominent way. I'll be showing you data where we've replaced this piece of skull over the olfactory bulb with a glass window surgically so we can watch what's happening on that dorsal surface of the olfactory bulb. And you'll notice that the bulb in a mouse is huge relative to the overall brain. It's about the size of the cerebellum. But don't make too much out of that because I'll also point out that the size of the brain is quite similar to the size of the tongue. So that's what the olfactory bulb looks like in a mouse. Here it is from the side, this kind of large structure that's kind of out in front in a mouse. But let's compare that to other creatures, right? So the olfactory bulbs in a dog. This is a view of the brain from the bottom. Here's a horse. Quite large, prominent olfactory bulbs, even as the rest of the brain is larger. But if you cut to a human, glance, you'd almost hardly see it, right? So this is the human brain viewed from below. If we zoom in the boxed area, here are the human olfactory bulbs, left and right. They're quite small. They are part of the brain, but they do physically seem to be separate. They're connected very densely with the nose itself. And then the olfactory tract is this nerve that comes back. That's not really a nerve, but this tract that comes back from the olfactory bulb into places like the piriform cortex and the amygdala. So this is quite a different size, certainly a different relative size than in the mouse. So here is the mouse with the same view, and you'd see the bulbs obviously seem enormous in comparison in the mouse and the human. That's actually the basis of the myth that Jurgis alluded to, that because the olfactory bulbs are proportionately really tiny in a human, it was sort of misunderstood to mean that that must mean that we can't smell very well. And so just as a demonstration, I took literally this olfactory bulb, this one that is depicted, and I put it next to this olfactory bulb that is depicted, just so you could see the relative size independent of the rest of the brain. And it looks like this. Human olfactory bulb is actually enormous compared to the mouse. And so this idea that it's too small to function is, of course, in retrospect, perhaps a little bit silly. But then there's also the question of why is it so big? Does it have many more neurons? Does it have a lot of synapses or fibers coming in from other places? Like what's going on there? And so I looked in the literature and tried to figure out, well, how many neurons are there in an olfactory bulb across species lines? And to produce this graph, and I'm not going to walk you through every Latin name for every species. The gist of it is that every dot here corresponds to data from the literature. I didn't collect any of this data myself, of just the number of neurons in an olfactory bulb corresponding to that animal. And this is a log scale. So this is 10 to the 5, to the 6, to the 7, to the 8. And let me just label some of these things with pictures so you know what we're talking about. So that's the number of neurons in a mouse, and then a rat, olfactory bulb of a bunny, a capybara, a macaque monkey, star-nosed mole, obviously a very different creature in terms of the role of smell in its ecological niche, but nonetheless, it's very similar in terms of the number of neurons in its olfactory bulbs. And this is a human woman. In fact, that's my wife. And so this sense that maybe it's not the size of the bulb determines the number of neurons, it actually seems that mammals seem to have about 10 million olfactory bulb neurons, regardless of the size of the animal. And why would that be? Well, it's probably because we're all exploring the same olfactory world. And so you don't necessarily need to have a whole lot more neurons if you're exploring the same world as a mouse or as a man. So I don't want to spend a lot of time getting into the data, mostly from other laboratories, about how humans actually perform on olfactory tasks. But I'll show you briefly some data supporting the general idea that how good we are at smelling something depends on the odor. And of course, the comparison to other critters also depends on the odor. So this is some data from Matthias Latzke's lab. Basically, in the same experiment, he had 12 humans, 4 or 5 spider monkeys, and I think 5 mice, all try to detect various odorants. And he just measured the threshold for detecting those. And what he found is that which critter is best at detecting on the odor depends on the odor. And so in some cases, the mice are better than all of the humans. This is the triangles is the range of all 12 humans, who are maybe similar to the spider monkeys. For this odor, all 12 humans were more sensitive to the odor than all of the mice and the spider monkeys, and so forth. And if you were to look across studies and just look at the published thresholds for different odors and many different species, you get a graph, or more to the point, Matthias got a graph that looks kind of like this. And so this is the threshold for detecting the odor. This is a log scale. Bigger numbers mean you're more sensitive. And every dot here is their published threshold from a different critter, from a mouse and a human to a dog or a vampire bat or a hedgehog. What you'd see as you look across just a really straightforward set of simple odors, basically just variance on carbon chain length. What you see is that for different odors, different animals are the most sensitive. And so I just circled all the mouse data, and here's all the human data for many odors on this particular sort of arbitrary subset of odors to look at. The mice is the most sensitive, but then for longer carbon chains, it's kind of in the middle of the pack. Humans are the most sensitive out of all the creatures that have been studied for 2 of these odors. I'm just trying to make the broader point that since I'm going to show you data from both mice and humans, that mice and humans both have a good sense of smell that makes a difference in our lives. All right, but there is one caveat, which is that I can't say if what I'm about to say is true for a mouse, but I can say for a human that our sense of smell is very changeable, right? It adapts very quickly, but also it's very strongly subject to influence by expectations. And here I particularly feel the fact that I'm not in Riga with you, because if I were there, I would actually be opening bottles of odors to show you some tricks. And there's a famous demonstration that actually was originally reported by William James, the sort of father of psychology, where he wrote, "We know that a weak smell or taste can be interpreted differently depending on how you label it." And so I have got a demonstration where I can give you artificial strawberries and you go, "Oh, that smells like a wonderful artificial strawberry," but it's actually banana, right? Or I'll give you an odor and I'll tell you, "Oh, this is, an artificial cheese." And you say, "Oh, that smells like a pretty good artificial, like Parmesan cheese." And then I'll secretly give you the same bottle later and I'll tell you that it's vomit. And I've gotten you to believe that it could really be vomit in the bottle. You will actually experience that same smell as disgusting rather than as the cheese that you might have put on your food 5 minutes earlier. So I think, and I don't have a lot of evidence, though I'll show you some bits and pieces today, I think that one of the things that larger olfactory bulb with its larger structure that probably has more neurons, fewer neurons, same amount of neurons, the larger number of synapses is actually neurons from other brain regions projecting fibers down into the olfactory bulb to communicate information about the rest of the world. And I will show you bits and pieces of that. So, I've just said that the olfactory system changes, perhaps that it particularly changes in humans. And that's actually a pretty risky, dangerous thing for a sensory system to do, right? If I gave you a microphone to record something, it would be a problem if that microphone was constantly changing its sensitivity, or if it was constantly changing how loud how the gain and amplified signals to, or what frequencies it was able to hear. It's kind of strange in a way to think that sensory systems change. So I want to start by talking about why that might happen. And so the easiest example is just to optimize your sensory system for a particular function or a particular environment or a particular goal, right? So pursue that the microphone analogy. If I were recording in a recording studio, recording a musician, I would be taking the raw output of the microphone and I would be putting it through various equalizers and sound processing systems to maybe isolate the guitar, isolate the voice on one microphone. And then I'd be using more equipment. I'm using some old-fashioned pictures here for fun, a mixing board to combine the different microphones to get the overall sound that suits my goal, right? So you might actually want to change a sensory system at one step in, even if you leave the microphone the same. Another goal I kind of already said, which is independent of sensory function, the ability to actually perceive, you might want your sensory system to change its code or change its activity in order to better link the perfectly detectable, perfectly understandable percept to things that you want that percept to evoke, to attract attention, to drive emotion, to drive behavior. And I kind of already alluded to that, right? Sort of disgust of a food that's rotten, or the attraction of food that smells good, right? If you could change your sensory system not to necessarily because you wanted to be able to detect the Cinnabon, but instead that when you smelled it, you would respond to it, you might want to change the sensory system to make that coupling more effective, right? So the question is, when would you want a sensory system to change? And there's a lot of complexity here. I'll give you just a taste, right? So of course, you might very well want to have adaptive responses, that the sensory changes, that's adjusting to stimuli as they come in, right? And the most obvious example of this is your pupil, right? Change the lightness or the brightness of the light in a room, and the pupil will expand and contract in response. That's a sensory system adapting, right? It's also the case that you can have a lot of value for having a sensory system change in an anticipatory way, right? And so if I had been there to do this illusion with you with the odors, that would be a priming based illusion. That me telling you this is strawberry, this is banana, this is strawberry, this is banana, that actually is sort of an anticipatory change. Your brain is anticipating what it's going to find, and if it gets something that's close, it kind of fills in the gap. But there's also another example I can do virtually here, which is that because you develop a cognitive model of the world, your sensory systems build in some expectation, and you may experience sounds, for instance, that aren't there. So here's a picture. Here's going to be a sound you've never heard from an image you've probably never seen. And I want to promise you, your speakers are not on. There is no audio in what I'm about to show you, but some of you will hear it. Not everybody, but some people hear a thump when that lands. Even though you've clearly never actually seen this in real life, you have no empirical reason to expect that there's a thump. Your brain anticipates it, and your auditory system sometimes can't resist putting the thump there even when it's not actually happening. All right? So then what else would you want it to change? Well, during ongoing responses, you might actually want to change your sensory processing to represent the world based on new information you have about it. So let me give you an example. Here's a beautiful piece of art. This is the Madonna and Child, of course. And what you perhaps have not noticed is that for some strange reason, Mary has something on her head. It's a little creature. It looks actually very much like a creature called Cookie Monster, which is a very popular character in American children's shows. And having now seen that Mary has got Cookie Monster on her head, you will continue. just see Mary with Cookie Monster on her head. In fact, I have used this illustration for a few years now. I cannot fail to see Cookie Monster. I cannot go back to my original perception of this art. And so this is sensory systems changing to build in information that you've got. All right. So what we want to do is to take that and ask broader questions about what can we tell from the way a sensory system's changed about what the person or the animal actually thinks is going to happen. All right. So in a sense, what I'm telling you is that sensory dynamics, the changes in sensory systems, they have a cognitive component insofar as they're building in pre-existing learned information about the world, either to better interpret ambiguous sensory information or to couple the sensory input to the corresponding behavior that would make sense. So that's kind of the theme of the data I'm about to show you. And just to kind of tie it all together for one last note, if sensory dynamics do have a cognitive component, what that means is that we should see beliefs about the world reflected in sensory processing, maybe even beliefs that are incorrect or that have no empirical basis for the organism. And then also, if you have cognitive dysfunction, you might be expected to see that dysfunction manifest in particular kinds of sensory dysfunction. And one of the last pieces of data I'm going to get to today is actually showing you some data from humans who have a particular kind of dysfunction, and we actually see changes that map onto that in their sensory systems. But first, let's talk about mice. So please excuse me, I have a very sore throat this morning. I'm going to do my best to keep going. So I'm going to use the mouse olfactory system as a venue to study the representation of sensory information from odors and see how it changes. And again, if I were in Riga, this joke would make more sense. But the first thing I like to do is to invite people to look up my nose, right? So you're not there in person, but this is actually my nose with an otoscope. And this pocket way up here at the top of the nasal passage, that is almost like a slightly yellow tinge in the membrane, or in the epithelium, I should say. That is the olfactory epithelium. That's the area in the olfactory cleft where odors are actually detected by your nervous system. And so that's mine. This is not mine, but mouse has the same sort of olfactory epithelium. It's got turbinate bones. And here you notice the shape of this, because you're going to see the shape again. From here, the sensory neurons detect odors, and their axons project back to the olfactory bulb. And the nature of that projection turns out to be really useful as an experimental system. So here's why. In the epithelium, if you took a cross section, you've got these individual sensory neurons in green here. They're not floating in space. There's other neurons holding them up, other non-neurons holding them up, I should say. And they've got dendrites that come up, and they have little cilia that make like a plate of spaghetti on the surface of the epithelium. And the odor comes in, and it touches the receptors in these cells on those cilia. And then they fire action potentials, and those signals travel down the axons and go back to the brain. So there's a huge universe of potential odor receptors, right? You've got 3 different opsons in your cones in the retina. You've got rods. That's 3, I guess 4. And because you've only got a few, you can only detect wavelengths between 400 and 700 nanometers of light. That's why you can't see infrared, can't see ultraviolet, because you don't have receptors that can cover that extra range. With odors, there's this vast universe of chemicals that could potentially come into your nose. And so we've evolved a very large number of possible receptors to be able to bind to basically this extraordinary range of physical chemicals that could show up. In fact, across mammals, including humans, the olfactory receptors is the largest class of genes in your whole genome. In humans, you've got about 390. In rodents, it's often about 100 different receptors. Details of that are a separate talk. But each of these cells in the nose picks just one of that repertoire of hundreds of receptors. And they're all mixed together in the nose. But when they send their axons back to the olfactory bulb, they map themselves out. They sort by receptor type. So if you look kind of in the background of this image, you see those turbinate bones again. This is the olfactory bulb, front edge of the brain right here. And so this is data where all of the cells that express a particular receptor, in this case, I think it was the P2 receptor, are colored blue. And you can see that there are little blue dots, little blue olfactory sensory neurons all over the epithelium in the nose. But as their axons reach the brain, they converge into this cool looking structure called a glomerulus. So this produces a mapping of odor receptor onto glomeruli on the surface of the olfactory bulb. Schematically, it looks like this. All the cells that express receptor A go here, all the cells that express receptor B go here, and so forth. And while this is a schematic, it's actually not far from reality. If I were to take a cross section through the olfactory bulb, it would look like that, where each set of axons coming in, they find each other and they dive in and form their glomerulus. Zoom in, you can see them. The axons kind of make a whole bunch of synapses inside. And if we step back to look at the whole olfactory bulb, this is the left bulb and the right bulb, we're looking down from above, you can see each glomerulus fairly distinctly. And so the reason I'm bringing this up is that it provides a really powerful tool for us to understand how the representation of an odor is mapped onto space at the input to the brain. And so in this case, every cell coming in from the nose has been modified genetically so that it releases, when it releases neurotransmitter, it lights up, it gets brighter. And this is not fMRI lights up. I mean, light physically comes out of it, which we collect with a microscope and a camera. And the idea here is that any given odor is going to bind to some particular subset of all those possible receptor types. And then it's going to drive input, neural input, to a corresponding subset of glomeruli. And in this mouse, those glomeruli will steadily get brighter as the neurotransmitter gets released from those cells responding to the odor. So we can look at this in vivo. This is one of these mice. This mouse is alive. It's got its head bolted to a microscope. It's getting sugar water and stuff to make it feel better about that. This is a live look. Well, not live now. At the time when it was taken, it was a live look into the surface, the dorsal surface of the olfactory bulb. And you can clearly see the glomeruli right here, the fibers coming in. Here's a glomerulus. Here's a glomerulus kind of under a blood vessel. See them, right? And so this is going to be a movie, the change in fluorescence that happens in this animal when an odor is presented. So there's a little bit of motion around the blood vessels. The odor comes on now. And you see a very specific set of glomeruli light up. This is not hard to see. One of the things I like to do with my students when they're getting started is I'll have them look through the eyepieces on the microscope, and you can just literally see the light come out of the olfactory bulb when an odor is presented. So this is really zoomed in so that you can see exactly what's going on in a few glomeruli. But for the purposes of the rest of this talk, it's actually really useful to zoom out and be able to see lots of glomeruli across the whole surface at once. So to do that efficiently, we use pseudocolor instead of grayscale. So look at this pattern. You're going to see it again. Here's that pattern. So now when we zoom out, we're going to be able to look at both olfactory bulbs at the same time while the mouse is smelling an odor. And you're going to see a movie here that's going to be pseudocolor. It's going to be the pattern of activity that happened, which glomeruli lit up during the odor presentation. So here we go. I hope. Yep. The odor comes on now. And you see a very specific set of glomeruli light up. It's roughly symmetrical left and right. And I'm going to jump out of the movie to a still image, right? And so that's the pattern that this particular odor, which was called methyl valerate, evoked. And now in the same animal, I think on the very next trial, we presented a different odor. And this is the movie of what happens with that odor. I think it's playing. Yep. Here's the odor. You see a different set of glomeruli light up. And so here's the still from that. So what you're seeing is different patterns of representations of the odor. And in fact, there's a cool, to further argue it's a representation. If you were to look at my records, you would see that the student who gave me this video to use forgot to label it. But that just stripe in this case is distinctive enough I know this mouse is smelling methyl valerate because that's what methyl valerate looks like. And I know that this is butyl acetate. That's what butyl acetate looks like. And so now you knew it. Now you know it too. If you see these stripes, you'll know that that particular odor in one of the data points in the talk was coming from a mouse that was smelling this fruity odor called methyl valerate. So the actual representations are a little bit more dynamic, right? Because you breathe and exhale odors. And so I'd show you a different way of visualizing the activity. It looks like this. This is going to be, both movies are going to play at once. This bouncing ball is going to show you the respiratory cycle of the mouse breathing in and out and in and out. And you'll see it's a very dynamic movie, but the overall patterns are going to be familiar to you. Breathing, there's a little bit of a blood vessel movement. The odor starts. You see those patterns. You see they're temporally complex as well as spatially distinctive, and they fade out. You want to see it again? I'll play it one more time. Sort of background activity, not much going on. The odor comes on, and you see this breath by breath pulse of particular subsets of glomeruli, different odors driving different glomeruli. So if you watch closely, I bet you would have actually recognized that this one on the left was methyl valerate again with those two stripes. And so you could now tell what odor the mouse was smelling by looking at its neural activity. So we've got a pretty cool ability to see what the brain actually is representing about the stimulus coming from the nose. And I want to emphasize that we're not way back in the cortex someplace talking about pattern recognition to try to find how activity maps to the stimulus. The data you're looking at from these mice, this was literally imaging from the nerve terminals, the presynaptic terminals of the olfactory nerve, where they're coming into the brain. And so the other end of the cells that are responding here are in the nose touching the odor molecule, and we're watching their output. And then here we're imaging from some postsynaptic neurons that are being driven by that synaptic output, and we're watching some cumulative metric of their calcium signals reflecting their spiking activity. Okay, I hope everybody's with me. So just as a quick aside, this code of glomerulus to odor is ubiquitous across species, right? It goes back to the dog. The Broca had it in the dog. You see it in monkeys and hamsters and bowhead whales and so forth. So now let's talk about how they can change. Let's take the turn and start talking about learning and maybe things that look a little cognitive showing up in the early sensory system. So here's the first part. What happens if an odor becomes common in the environment, right? If you were designing a sensory system, it might be useful to have the system recognize the natural statistics of the environment and tune itself so that it has sort of an optimum code that's the most efficient way to represent that. And so we can do that in these mice. We can change their olfactory environments and see if the system adjusts. So here's a map to an odor. In this case, as you might recognize, it's methyl valerate. And then this is the map to a different odor. This is butyl acetate, which smells like banana. I'm sorry, butyl acetate, different pattern. So, sorry, actually almost banana. This is a baseline before we did anything to these mice. And then we took the mice and we let them live in a chamber where for many hours a day, an odor was pumped in for a few hours and then off, hours on and hours off. And what you'd see is that there's a great reduction in the number of glomeruli that seem to be activated or to be getting input from the nose in this case for the odor that it was exposed to. This is not simple adaptation. It persists for hours after they come out of the box, at least, perhaps days. And it seems to be somewhat odor specific, right? You see a decrease here that's more or less unchanged for another somewhat similar odor in the same animal. And if you were to look across odors, there's a big decrease in the number of responses you'd see for the odor that was in the environment, some reduction for a similar odor, and no change for other odors. Now, this by itself maybe doesn't look all that cognitive. Yeah, it's natural statistics, but it feels a little bit like adaptation. I'm going to show you in a few minutes the consequences for this animal perception. But first, I'm going to ask something that looks a lot more like learning and memory. We're going to do something that's just classical fear conditioning. And in fact, we're going to do discriminative fear conditioning. So the mouse gets put in the box, and it gets methyl valerate blown in, and it gets a shock. And then a few minutes later, it gets butyl acetate blown in for a few seconds, and it doesn't get a shock. And you interleave those a few times for a few days, and you see what changes. And so is called discriminative fear conditioning, where we've worked at it to make sure the mouse is afraid of one odor and not another, right? And then here's what the maps do. So I'm going to direct your attention at the bottom. This is the control odor that was pumped in but wasn't associated with any kind of shock. And in this case, you see no change before to after conditioning in the same animal. But for the interleaved odors that did predict shock, here's the response at baseline, and here it is after 3 days of conditioning. Now you've got a larger response, somewhat broader response, depending on how you define it. This is a signal that's coming out of the olfactory nerve, right? These are the neurons that are transducing the odor. This is analogous to seeing the effects of fear conditioning, odor-specific effects of fear conditioning. Imagine this was in a rod or a cone, right? That's where this is in the sensory system for the olfactory system. It's the very first neuron in its presynaptic terminals. You can see the effects of learning, right? And just to show you this, the distributions, because I'm not a big fan of bar charts, even though I use them quite a lot in this talk. This is the distribution of all the glomerular responses to all the odors in the panel across all the mice. Black is before conditioning and red is post-conditioning. Control odor that didn't predict a shock. And here it is preconditioning, and then in red post-conditioning, the odor that does predict a shock. Again, this is every data point from this particular experiment. All right, so that's discriminative conditioning. And the thing is about this paradigm, when the mouse becomes scared of one particular odor, it's right. That odor does predict a shock. It is taking a correct understanding of the world. But what if instead we give the mouse an ambiguous situation where the mouse thinks an odor predicts a shock, but in fact it's wrong, or at least it hasn't had the experience to know for sure. And so we change the paradigm. It's the same mice, but we change the paradigm so that it's ambiguous. There's one odor, not two. There's a big shock. There's only 1 day of training. And we design a paradigm to lead the mouse to become afraid of lots of odors that it's never experienced before, not just the one that was actually presented in the box. All right. One odor during training, that odor, in this case, it varied. I think for most of our mice, it was methylvalerate again. And behaviorally, if you look at their conditioned response, the percent of time they're freezing, these are a series of different odors, some similar, some quite different in their smell. The mouse is, if anything, slightly more afraid of these other brand new odors it's never smelled before, having had this generalizing single odor conditioning. They're scared of lots of stuff, right? So if we then move on to what happened in the olfactory system, and again, we're looking at the output of the nerve in this particular experiment. These are the baseline responses to those 4 different odors. You see 4 different patterns. And then after this generalizing fear conditioning, of course, you see the increase in response for the odor that did predict shock, but you see similar increases for all the other odors the animal has also become afraid of. We think that's really interesting because these are changes happening in sensory neurons in response to odors that they've never smelled before. But based on the mouse's belief that these odors are scary and should evoke a fearful response, you see this change even as early as the nerve terminals. And then you'd say, well, maybe this is some kind of feedback. The mouse gets the odor, it becomes scared. It's anesthetized for this imaging for the control for that. But even if you're not happy with anesthesia as a control, if you look at the waveform here, which is using that faster metric of response, you can see even on the rising phase, the first inhalation of the scary odor can tell that it's the scary odor in red compared to its own baseline in white. So these are really rapid changes that seem to be sitting there waiting for the odor to come along. So you get a facilitated olfactory response for some reason. So then the question is, or the tangent, how the heck could the olfactory bulb ever know that an odor predicts a shock, right? A shock is a somatosensory thing. We're in an olfactory system. Well, it turns out, even if you go back to Golgi from 1875, first Golgi stain ever presented, the first sort of study of individual neurons was in the olfactory bulb. And you'll recognize the glomeruli up at the top below the blood vessels. These are called mitral cells, sending their axons off to all sorts of places all over the brain. But all these fibers coming in are actually coming from other places in the brain down into the olfactory bulb. If you look at it with modern techniques, you often see individual glomeruli have these shells of neuromodulatory or cortical inputs coming down positioned to influence what's happening even in this earliest representation in the glomerular code for the odor. And it's even true that if you make big changes in the state of the mouse from like an arousal or a neuromodulatory perspective, you can actually get quite large changes in the output nerve. A completely anesthetized, completely unconditioned mouse, just blocking the amygdala, shutting down the amygdala, causes changes in the odor release, odor-evoked release of neurotransmitter from the nerve. All right, so we're getting somewhere. But these descending information and descending fibers carry information that the mouse got shocked. It can. You're about to see almost the first people in the world to see the response of mitral cells in the olfactory bulb to a tail shock. And here's a different population of cells, periglomerular interneurons, responding to a tail shock. A lot going on. So somehow, we're still figuring out the circuit, but somehow information about global modulatory states and possibly specific signals like pain are reaching the olfactory system. And I'm going to quickly just kind of show you imaging different cell types in the bulb, see different patterns. Everything except sensory neuron terminals themselves seem to know about the shock. All right. And it evolves over sniffs sometimes. After a shock, you see some signals that evolve. So let's move on. What does this actually do? Right? We have a sensory system changing. We said that could be dangerous. It could be useful. Let's find out what it's for. So we're about to show you some perceptual changes. So if you take a mouse and you put it in a box and you have a little filter paper with an odor on it, and you just put that filter paper in the box over and over again, the mouse will eventually goes over and investigates a little less, and eventually it ignores it. And if you were to change the odor, go like hex, 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 to a similar but not exactly the same odor, if the mouse thinks the odor hasn't changed, it will continue to investigate it less. But if you switch it and the mouse can tell the difference, it will actually start investigating that odor more because, hey, that's new. So this is an experiment where we had the mouse being put in that stinking environment like we talked about, and had that remapping of the peripheral input based on natural statistics. And when you do that, you'd actually see the representations become a little more different. And lo and behold, those mice become better able to act as if when you switch the odor to a similar one, they go and investigate it. They perceive the change in ways that control mice that had the same experience act as if that odor was the same odor that the last time. So you got a small improvement in the ability to tell odors apart. And if you try to take this out to a more abstract space, if you say, well, there's all these glomeruli, and as a group, they represent the sort of pattern, the pattern represents the odor that's out there in the world, you could do things like principal components analysis and develop the idea of a trajectory that shows the state of the network over time. So an odor comes on, and the odor kind of the pattern of the network kind of drifts off to a different space. And in one odor with one pattern of glomeruli, you'd go one place in the space, maybe over here. A different odor coming on, you'd maybe go to a different place in this multidimensional space. It's a way of taking these pictures of patterns of responses and putting them into a shared axis where you can see them change over time and you can compare. So this is odor presentations, 4 of one odor and 4 of another, illustrating that you can use this technique to visualize how different the representations are and how they diverge over time. And so if we use this technique to see what's happening in those representations in the discriminative fear conditioning or in the single odor generalizing fear conditioning, you basically see that when the mouse is becoming more scared of all the odors, the representations of those odors in multidimensional space converges. It's as if things become less different in the neural activity and also become less different to the mouse. And of course, the behavioral responses also converge. By contrast, if you look at it in the mouse that learn to discriminate, one odor is scary, one odor is not, actually the patterns become more dissimilar. The timing is that they become more dissimilar, especially at the beginning of the odor presentation, when it's most important to know that, oh, this is the scary odor or this is not. So we think that there may be a perceptual or some kind of discriminative response representation in the glomeruli that's paralleling the discrimination or convergence of representations that's seen sort of perceptually and behaviorally. So that's the mouse data. And I've shown you a few things. I've shown you that changes in odor representations can manifest as modest differences in odor perception. I've shown you that discriminative conditioning causes odor representations to become more different. I've shown you that generalizing conditioning causes odor representations to become more similar. That these things happen as early as the olfactory nerve terminals. So literally the neurons that are detecting the odor are at least aware in some sense of the change. I don't know that they're learning, but they seem to be being informed of the contingency. We actually know that there's a presynaptic modulation via a GABA B receptor that seems to be part of the mechanism of communicating the learned information back into the olfactory nerve terminal, but that's beyond the scope of this talk. So that's the mouse. What about people? Is there a way to figure out in people where we can't image glomerular patterns, MRI can't do it, and we can't do all these experiments, but are there things we might be able to check in a person that might tell us that the same sorts of mechanisms might be happening? And so, yes, we kind of head fix our humans the way the mouse was head fixed under the microscope. We use the same olfactory apparatus, and we actually set up humans to smell an odor and get a mild shock on their wrist and smell a different odor and not get the shock, and use a similar sort of paradigm to what we use on the mice. And if it were true that we would be causing neural representations or perceptual representations to converge and diverge, in theory, we might be able to see changes in human perception that would map onto the sort of differential meaning that people learn about the odors. And so I'm not going to walk you through all the experimental details, but I'm going to show you the bottom line. If you give a human two odors, and in fact, it's the same pair of odors that I just showed us using in the mice. So hexaldehyde and heptaldehyde, there's one extra carbon on the carbon chain. We correct for differences in vapor pressure and stuff. So they're very similar. And when most people walk into the lab, we ask them to tell them apart on a forced choice test with 3 options. They're on average a little better than chance when they come in, but it's hard, right? Occasionally we get some people who can do it when they walk in the door, and then we exclude them from this part of the experiment. But for people for whom it is hard, it is quite hard. They're only a little bit above chance. And then we put them through just 40 minutes, I think it's 8 trials of each, of the odor presentation with one odor. Hexaldehyde gets the shock, heptaldehyde, and then it doesn't get a shock, or vice versa. We mix up which odor is which. And you can see that just that small change, small experience, makes them meaningfully better at telling the odors apart when we test them again on just, can you tell the odors apart afterwards, independent of any kind of shock during the testing. And then we also are measuring things like skin conductance responses, which I'm not going to have time to go into. And you can see that at the beginning, their skin conductance responses to the 2 odors are the same, and small. But then as the conditioning goes along, they not only become able perceptually to tell the odors apart much more reliably, but they also begin to show differential skin conductance responses, that they're afraid of one and not the other. We don't know which is the chicken and which is the egg, but they co-occur. They don't generalize, they discriminate between the odors after initially finding them close to indiscriminable. And so that's cool, but this is what I just said. The folks who have normal levels of trait anxiety do what I just described. That's most people. But remember, I said that if you had a dysfunction, cognitive dysfunction, you might expect it to manifest as a sensory dysfunction. What about the folks that come into the laboratory who are really anxious as a trait anxiety? So they were anxious before they ever got here. That's independent of the fact of being in the lab. That's called state anxiety. Trait anxiety is how anxious are you as you go about your daily life? Is anxiety a problem for you? Unfortunately, a meaningful number of students who walk in the door from just generic recruiting on campus actually score at a clinically relevant level of anxiety on some of our anxiety tests that we administer when they arrive. If you look at them before conditioning, they're no different at telling the odors apart. They find it equally difficult as the non-trait anxious folks. The high trait anxious folks are quite similar. And then the question is, when they have this aversive conditioning experience, what happens? Do they, like the normal the folks with healthy levels of anxiety, do they have that increase in discrimination perceptually? The answer to that is no. The folks who have sort of normal levels of trait anxiety normally show an improved discrimination between the odors. The folks that have really high levels of trait anxiety do not get better at telling the odors apart. And if you look at their skin conductance responses, they become afraid of both odors. They get a generalized fear response, where in fact they go from no particular response to either to a big response to both, right? We've got a situation where we've got generalizing fear in highly anxious folks that's correlated with a lack of change in their sensory systems that normally would have occurred. If you're curious about what happens if we were to reverse it, this is just, I don't have time to do the whole experiment. This is data across individuals. This is the improvement or decreased performance on the sensory discrimination task. People who got the conditioning get better. But if you look at the folks who had healthy trait anxiety that showed this improvement, and then you extinguish it and you just go odor, 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 odor with no shock, you actually see some reversion, but not completely. And so at a point where the skin conductance responses have completely extinguished, you actually continue to see this improved discrimination between the two odors. So we think it's actually the discrimination drives the skin conductance response, not the other way around. Okay? So, picture, I'm almost out of time. Big picture. Normal functioning of the olfactory system includes learning-induced neuroplasticity. It can happen as early as the nerve terminals. It is associated with perceptual changes in smell. We seem to see that in humans and in mice. Plasticity seems to reflect the individual's learned beliefs about the odors, whether they think the odor, this odor means shock, then you see the changes in the neuroplastic changes only for those odors. If they think lots of odors mean shock, then you see those changes for all those odors, right? Independent of what the animal actually experienced, even for odors they've never smelled before. Then the absence of this aversive learning-induced perceptual plasticity in folks with high levels of anxiety, we call that a sensory dysfunction, that learning isn't inducing the normal changes in sensory processing. And so I'd say that supports my suggestion earlier that cognitive dysfunction can manifest as neurosensory dysfunction. And so the takeaway from all of this pile of data at the end of the talk is that humans and rodents, we're all exploring the world together. And in both of us, our sensory systems are constantly changing for various purposes. So I want to acknowledge the students who actually did all the work here. Michelle and Marley, who have both gotten their PhDs to move on to postdoctoral positions or various roles. They collected the fear conditioning data. Cindy Fast, seen here, she's now the Director of Operations for APOPO, the organization in Africa that trains rats to find landmines. So she collected the data on the amygdala and the circuitry connecting down to the olfactory bulb. Keith Perkins is the one with the cool data from the tail shocks causing activity in the olfactory bulb. He'll be publishing that soon, I hope. And I want to thank our funding agencies, and I'll take questions.
Thanks a lot, John. That was really, really awesome. Thanks. That was a absolutely cool, wonderful talk. And well, there is time for questions and comments. And yeah.
Happy to.
So there are a couple of thank you messages in the chat. But so
Yep.
are there any questions in the audience? While audience is thinking, I'm just curious. I mean, the one side of the story is, of course, that we are incredibly adaptable in terms of our olfactory perception. But well, are there any evidence that there are some innate, let's say, kind of olfactory constancy? I mean, there is this discussion that small kids are more sensitive to sweet odors,
Right.
but so I mean, is there the other side of the coin that there is some olfactory constancy in higher primates?
Yeah, it's a good question. There's there seems to be plasticity all the way down. You can actually see there's a the sensory neurons in the nose are constantly dying off and being replaced from a local population of stem cells throughout your lifespan. And you can actually see experiences related to what odors you encounter, or amount of rodent encounters, and what odors mean in terms of pairing with shocks, pairing with cocaine, things like that, actually biases the expression of odor receptors so that you get a change in the mixture of odor receptors neurons in the nose. And so there's a sense in which you've eliminated any purely bottom-up signal at all at that point, because you've literally got a different receptor base to work from. Nonetheless, you still have a remarkable constancy perceptually of quality. And so you might have noticed that none of the changes I showed you were like turning methyl valerate into butyl acetate. It tended to be sort of a scaling and a change in the sort of representational distances between similar odors or different odors. And if you thresholded that, you'd see things would become radically different. But it seems like the underlying sort of relative patterns are still there. And so you may be tuning or biasing, but not wholesale rewriting, unless perhaps the circumstance, if you were in a car accident and your olfactory nerve got cut, then there is sometimes a parosmia that develops as the system rewires and the old code isn't valid anymore and the brain doesn't understand what it's smelling and everything smells wrong. That happens to some folks after COVID-19 as well. But I think, to be completely honest, I think that the purpose of most of piriform cortex, the sort of olfactory cortex that's getting a lot of this information, and perhaps some of the purpose of the loops between cortex and bulb, is to build a structure that's like an auto-associator or a self-organizing network. All of the synaptic plasticity features you need seem to be there. The recurrent connections you would need seem to be there. And so I suspect that what's happening is that the percept is emerging at the level of the autoassociator, and it's basically able to get more or less quality information, but it's probably doing some measure of pattern completion. It's probably doing some decision about to separate or to merge patterns. And so for the humans, for instance, who are learning to discriminate hexanal from heptanal over the course of 30 minutes, like they're not growing new odor receptors, they're not wiring new neurons. What I think is that the system was always able to discriminate, but that the higher level sort of category generator or autocompleter, or however you want to, whatever language you want to use, is sort of deciding, oh, wait, I've got one representation for two different things, I need to split them. And so I bet if I could see what was happening in piriform cortex, you'd see a segregation emerge. And actually, Wen Li and Jay Gottfried had a paper some years back using fMRI in humans doing a somewhat similar paradigm. And they did see a divergence in sort of global patterns of odor-evoked activity in piriform as you learned to sort of tell enantiomers apart. So,
well, thanks a lot. Thank you. So, any questions? Any questions? Is it okay, Anita, yeah, go ahead.
Yes, thank you so much for extremely exciting and fascinating talk. I don't really have a question specifically regarding to the topics you covered today, probably because I still have to process all the information. So
sure.
I can't approach the current subject. But can I just use this opportunity to ask about your opinion, if it's not stretching too far out, about pheromone perception in humans?
Sure. So it's a question that sometimes requires unpacking because different people have meant different things by pheromones over the centuries. I subscribe to the belief that we need to stick to the sort of original definition of a pheromone, which is a hormone delivered by a conspecific, another animal of the same sort, that induces an effect, right? If you have adrenaline that gets released in your body, your heart doesn't decide whether to speed up or not, right? And so if you asked me to give you an example of a human pheromone, the honest example I would give is an EpiPen, right? When we literally inject another person with epinephrine to cause a hormonal change. And I would say that if I drop a stink bomb in an audience, people will go, oh, that's terrible, and might leave. And that's a behavior that's evoked by an odor, but I wouldn't call that a pheromone for the same reason that if I shouted fire, they would also jump up and leave, and we wouldn't call that an audio mode, right? That's I credit that idea to Dick Doty. It's a great example. And so it's absolutely true that in some animals we've identified specific chemicals. Benoit Schall's got a chemical he identified called 2-methyl-2-butanal that's expressed from the undercarriage of a lactating baby rabbit. And if you blow 2-methyl-2-butanal at the nose of a really young, like newborn rabbit, it will suckle on the machine that blows the odor out, right? It's a really clear thing. There are scent trails that will make moths you can completely control the moth by where you put the odor, right? Those seem to be sort of pheromonal, whether they're technically hormones, endocrine things, I'm not so sure. But there's clearly a specific chemical that does it. And no such chemicals have been observed in humans. No compelled behavior have been observed in humans. And so if we take a strict definition of pheromone as I think it should be interpreted, then no, I teach this in my classes. There are no documented human pheromones. I will give you one piece of historical commentary, which is a little complicated. So dogs and rodents and a lot of other creatures have an additional sensory system that we don't have. The reason, like if I were to say, I'm going to give you a glass of wine, smell it. 60% of the time, the people who I ask say, I would take one long, deep sniff. And about 40% of the time, people say, I take a series of fast sniffs. But nobody ever says, I'm going to stick my nose in the wine, right? But a dog sticks its nose in the wine. It sticks its nose on the fire hydrant, right? And that's because it has a system that you don't have called a vomeronasal organ. It's like a tube in the nose. That's a pump. And its job is to suck non-volatile or barely volatile liquids that are chemicals into the tube. And there's a special nerve called the vomeronasal nerve. It has cells that express a special set of receptors. We don't have the receptor genes. We don't have the nerve. It projects back to a special brain region that's called the accessory olfactory bulb. We ain't got it at all. And so we don't have that system. Historically, it's been believed, going back 40 years, that was the pheromone system. And that the reason humans didn't have pheromones was because we didn't have a vomeronasal organ and an accessory olfactory bulb. And we didn't have the hardware, so we don't have the pheromones. That's actually not true. If you take the pheromones in animals that are really actually seem to be pheromones in the traditional sense, the regular olfactory system also responds to them if they're volatile, right? And not if they're not volatile. And so I think it's actually a better description. We need a better word. I almost think you could describe the vomeronasal system as kind of like nose taste, because you have to actually make physical contact with the source, right? As opposed to just getting it in the air. And then if we think of that as this whole parallel sensory system for non-volatiles, and we don't have that maybe because we walk on two legs and our noses are way off the ground, then I think it's easier to say, well, in the same way they have a different complement of receptors in their noses than we do, they also have a different complement of receptors for low and non-volatile liquids than we do, because they evolved to a different ecological niche. So I don't think it has anything to do with pheromones. I just think that pheromones might have non-volatile components.
Thank you so much. I feel
You're welcome.
I just heard another lecture.
Sorry, I'm like a jukebox. I'm like a jukebox. You hit the pheromone button.
Well, thanks a lot. So any final question from the audience? Any final remark? If there are no.
Okay, I will also ask just one question short.
Sure.
Thanks a lot. It was really really exciting talk. And the question is about conditioning. If there would be positive conditioning, would the same pattern occur, or what could be the result?
Yeah, it's a question that I've tried to answer. The short answer is yes, but the data is not as compelling as you'd hope. And the reason is it's very hard to make an experience that's as positive for a mouse or a person as a shock is to a mouse. And so you don't get the same immediate obvious changes, but you can get some. So in the mice, a colleague of mine used cocaine, paired odors with cocaine injections, and did find that there was like a remapping of the epithelium. If you did this a lot for like a month, you could get more receptors in the nose that were sensitive to the odor that predicted the cocaine. And I had a student for a while, sort of an exchange student who was only in my lab for a brief period, who tried to do it with like sugar water rewards. And you have to work a lot harder to get the mouse to necessarily understand that it's the odor that predicts the sugar in that case. But she did, and for the 4 mice that we got data, it was really convincing that you did get a similar change. But we only ever got 4 mice worth, and the student's time in the United States ended, and that was the end of that project, unfortunately. We've explored doing it in humans. In fact, I had, I guess I shouldn't put a name on this story. Someone from a perfume company approached me once, asking if we could maybe come up with like some really attractive experimenter that gave the subject a hug while wearing a perfume, and would that improve their discrimination between odors that were similar to or different from the perfume. And I said, I'm not doing that experiment. So we've talked about doing something with maybe chocolate, or trying to get some kind of positive with humans. But the problem is pictures of fuzzy bunnies and cute cute babies and fuzzy bunnies don't evoke a very strong response for long. So it's not clear if we would expect that to produce a change. Anything that uses, sugar or desserts and stuff, you're introducing a metabolic confound and a taste confound. So I do think it could be solved, but it's harder than to just give some people some mild shocks. And then there's a much more practical reason, which is a lot of my work is funded by the National Institute on Mental Health, because we've got this idea that part of what's happening in anxiety is a physiological inability to learn to discriminate between potentially things that are threatening and things that aren't. And we actually now have data that folks who are anxious are in fact different. And so the practical goal of the sort of short-term work is to move towards a model that's more like responses to trauma and sort of aversive stimuli, as that seems like the highest priority for us, in addition to being the most experimentally tractable.
Thanks.
Yeah.
Thanks a lot. So there is a short, final question in the chat by Ant. So is asking.
I see it. Yeah, I see it. So the question about highly sensitive people, I have to wrestle with that a little bit, because there's 2 senses in which that could apply. So remember at the very beginning of the talk, I had the slide of the guy who was cutting onions and the sort of response you get. I sometimes argue that if we just found the olfactory system and we didn't know about smell, we would think it was part of the immune system, because it's detecting things that are physically coming into the body. And there's a chemical detection system that's sort of related to olfaction that detects damage, it detects potential infection, and it actually causes local plasma extravasation. So plasma comes out of the blood vessels and your nose starts running, right? You get mast cell degranulation, you get inflammation in your nose. That's why things like an onion can actually make you have this feeling that's kind of like, having a head cold. It's your sensory system detecting the chemicals and reacting accordingly. So if when we talk about smell kind of colloquially, we're including that chemical detection, then I would say for sure there are likely differences in how strongly people react to the trigeminal mediated chemosensory component that to me is part of the olfactory experience. And for sure, different people have different degrees of adaptation with that. People that eat spicy food are much less sensitive to spicy food, in part because that system has adapted. And that is related to this sort of sense of like burning from an onion or the burning smell of ammonia if you smell a cleaning product, right? But almost every odor in the world has some activity in that parallel sort of agent detection. Again, it's almost like the immune system is screening everything in the nose. And so in the literal sense of sensitivity, I think you're probably right. Now, if I remember correctly, Elaine Aron's got a broader meaning of sensitivity, right? The idea of being influenceable by outside forces, including empathy from people outside of you and things like that. And there is some variance in our ability to respond, how we respond to other people's body odors and the impact that other strongly odored environments have on us, the degree of physiological arousal, what kind of skin conductance responses you get, what level of anxiety you're at. And in the same way that having like pleasant music playing can relax you, being in an environment that smells in a way that has maybe associated with pleasant smells. For me, it's the smell of a pine forest is relaxing to me, in part because that's where I spent my happiest days of my childhood, right? And so I do think there is a broader sense in which odors provide kind of a background that influences our emotional state. It influences the comfort with which we interact with others, perhaps. And so while the classic aromatherapy, smell ginger for alertness stuff has very little, not none, but very little actual scientific experimentation attached to it, I'm inclined to think that there really is a modulatory role that underpins a lot of other things.
Well, thanks a lot. Thank you so much. So I think on this note, we might say thanks a lot to our speaker again. This was really, really awesome, cool, wonderful talk. And this is not just on behalf of our lab, but also on behalf of University of Latvia and the Riga Study Engineers, our collaborators. So thanks a lot, John, and you are very warm welcome to Latvia at some point. We would be very delighted to see you in Riga.
I'd be delighted to.
Thank you
thank
so
you
much.
very much for inviting me. I appreciate the kind words. So.
Thank you so much. Thanks a lot. Thank you.
All right. Bye, everybody.
Have a nice day. Bye.
You too.